All right, for those who actually tried to watch my first recording of this, I'm sorry, I guess my camera's not working, so I've got a camera that's over here on the side. I can't change that. You probably don't need to see me anyway. But that particular recording, I have no idea how, but it's flipped upside down and backwards, and I was unable to fix it. So I'm recording this. So again, this is part of Lecture 3. A part that we did not cover in class and won't cover in class. Hormonal control of catabolism is the primary lecture, and we went over some things in class. We spent a lot of times pertaining to the article, and so I didn't want to, I wanted to make sure that time was spent in class. But what this lecture is really about is the metabolic pathways that are involved with catabolism. Although I'm going to admit there is one pathway we're going to cover, that is anabolism. And that's okay. This was just a convenient place to kind of put that in. And so I'm going to go through this, and hopefully this works this time. Right, our first pathway is going to be lipogenesis. And I've got it actually in a bullet point down here at the bottom. But we talked at the beginning of this semester that lipogenesis is actually a slightly unclear term. It can be used in more than one way in the literature. And so I'm referring to lipogenesis as including both fatty acid synthesis and triglyceride synthesis, both of which are separate pathways, but of course intertwined in some ways. But in general, lipogenesis, including both fatty acid synthesis and triglyceride synthesis, are stimulated by insulin. Remember, insulin is a storage hormone. This is lipogenesis. We're creating fat. That is the role of insulin. This gets inhibited by 3 different hormones, and these tend to kind of group, especially regarding metabolism. They'll each have different separate effects outside metabolism. But glucagon, epinephrine, and glucocorticoids, and we talked about where these come from, or we'll talk about where these come from in the lecture. Lipogenesis takes place in the cytoplasm. Almost all of these processes will except 2. So just remember that. Everything's in the cytoplasm, except when I tell you otherwise. So that's kind of a rule, so you don't have to memorize each one. And lipogenesis, its primary location is the liver, because it's doing both things, fatty acid synthesis and triglyceride synthesis. It doesn't mean it's the only place, but it is a primary place. Mammary gland needs to do this as well. Kidney can do this. Most of these steps also occur in adipose tissue, but the reason they don't is because we don't synthesize fatty acids to any great degree in adipose tissue. The fat that is delivered to adipose tissue is already made most of the time, and we actually pull it apart and put it back together. And so we're not doing this to a large degree in adipose tissue, but we do it to some degree. Some of these steps are taking place there. All right, so fatty acid synthesis first. And so we're going to start back up here. Fatty acid synthesis, usually we start with acetyl-CoA, but I just want to remind you that acetyl-CoA can come from pyruvate, right? If you come down glycolysis, in the presence of oxygen, glycolysis, the end product is pyruvate. And for that pyruvate to continue on, so we can continue to extract all the energy that we can out of the original glucose molecule, it needs to become acetyl-CoA. Right? And so here we have acetyl-CoA. That's our starting molecule. And acetyl-CoA can be converted to malonyl-CoA, and that's done by an enzyme called acetyl-CoA carboxylase. And we have mentioned, of course, this before. Acetyl-CoA carboxylase, note, in the presence of insulin is stimulated. This doesn't mean insulin goes in and directly stimulates it, right? It's insulin binds to its receptor. It activates a signaling cascade, and we've learned in lecture, right? PI3 kinase AKT, the same signaling cascade, and it's going to end up in activation of acetyl-CoA carboxylase. We're going to crank its activity up way higher so that it can convert more acetyl-CoA to malonyl-CoA. The same enzyme is inhibited by glucagon and epinephrine. While we release glucagon and epinephrine in an unfasted state or in a fight-to-flight state where we need to mobilize energy, we don't want to be trying to store energy during those times. And so glucagon and epinephrine both inhibit insulin. All right, this step is generally considered irreversible. and it requires ATP, which is one reason it's irreversible, right? You've got to put energy in, you'd have to have that energy re-extracted, right, to get it back out. A little more than we want to go into. In fact, there's actually a second acetyl-CoA carboxylase enzyme to try and complicate this more. We're just going to stick to this one. All right, our second step is we take malonyl-CoA, which is a 2-carbon molecule. Acetyl-CoA was as well. We just changed a couple of things. And we're going to keep adding 2 carbons to it, right? Here's 2 more carbons, and then we add 2 more carbons. You get the idea here, right? And we keep adding 2 carbons at a time. This creates a fatty acid. Fatty acids are essentially carbon chains. Now, another name for a fatty acid, just so you know, is an acyl group. So if you see this word acyl, think fatty acid. Acyl doesn't tell you the length. It just tells you, hey, there's this carbon chain, and it's a fatty acid, right? So it adds 2 carbons at a time, and that's regulated by this enzyme, fatty acid synthase. Oh, incidentally, I was just also going to point out before I leave this slide, because once I leave, I would have to re-record the whole thing. Excess calories, excess carbohydrates increases the amount of malonyl-CoA, forcing more of this to go this direction, right? It's simply a mass flow issue. This isn't water behind the dam and we're activating the process of the water going over the dam. This is the water's flowing, and so it's moving in a certain direction. The more water there is, the more it's going to move. All right, our next part of lipogenesis was triglyceride synthesis. Now remember, a triglyceride is a glycerol molecule, and I'm going to draw glycerol like this. I'm not going to label this. You can do that on your own. That's glycerol. And we are going to add 3 acyl groups or fatty acids to this molecule. And we're going to do them one at a time. Okay, we're going to do a fatty acid here. We're going to do a fatty acid here. We're going to do a fatty acid here. And then we will have a triglyceride. Well, where we're going to start is glycerol 3-phosphate. Right? So here's that glycerol molecule in the 3rd carbon position down here. We're going to have a phosphate group. Incidentally, let me find some notes here. I forgot to tell you a couple of things that were not in the slides on those first couple of slides there. So I'm going to interrupt myself here before we talk about glycerol 3-phosphate a little bit more, because what I want to tell you about each of these processes is what are the rate limiting steps. Now, I did not include a rate limiting step for fatty acid synthesis, actually, so we don't have to worry about it, and that's the only one we've talked about. But for triglyceride synthesis, we're going to include that. So now I've pulled this out, so I'll make sure and get this. All right, so we're starting with glycerol 3-phosphate. Well, where does glycerol 3-phosphate come from? It comes from glycolysis. Glycolysis, right, we have intermediates, right? We start with glucose, and then it's converted to this, converted to this, converted to this, and eventually to pyruvate, or in the absence of oxygen, lactate. Each one of those steps creates an intermediate. One of those intermediates is glycerol 3-phosphate. So the glycerol actually is extracted from glycolysis, if you will. It's created. It does not have to continue down to pyruvate. It can be shuttled over to this side towards making triglycerides. So here's the glycerol 3-phosphate, and we are going to bring in a fatty acid. And when you ever see this symbol here, right, this is a reaction. It's becoming something else. And when you see something like this coming in on the side, this is being added into the reaction. If you see something coming out, that's coming out of the reaction, right? That's actually another product. We draw these, what I would describe as more of a physiological pathway, a little bit different than you would write a chemical equation, which would include all of the reactants on one side and all of the products on the other. When we draw it like this, what we're showing is what we started with and what we ended with. And then on the sides, hey, we also added this and we also got this out of it. But ultimately, we're more interested in, hey, what did we start with and what did we end with pertaining to our pathway? All right, so here we started with a fatty acid, and we're adding a CoA, which creates an acyl CoA, or you could call this a fatty acid CoA if you would like. They're the same thing. All right, now we have this glycerol 3-phosphate, we have this acyl CoA, and they are going to interact right here, right? And they are going to form a monoglyceride. It's going to kick the CoA off in the process. So now we have a monoglyceride, right? We've added up here the first fatty acid. And that's where we're going to end. And this is technically known as an acylglycerol 3 phosphate. Right? You can see that in acyl glycerol 3 phosphate, right? So we've got that. Another term for this is a monoacylglycerol, or we mentioned a monoglyceride, so monoacylglycerol, MAG is the abbreviation. Well, now we're going to take another acyl-CoA. We got it from the same place, right? Fatty acid added CoA. We've got an acyl-CoA group that's going to interact with this monoacylglycerol. We're going to kick off a CoA. Incidentally, we also kick off the phosphate at the same time. I didn't draw that in here, but we got rid of that phosphate group. And that leaves us with a diglyceride. So the diglyceride, now the phosphate is gone, right? But now we have here's the glycerol molecule, and we have 2 fatty acids attached because we just attached another one. So that is termed again a diglyceride, or to be more technically accurate, a diacylglycerol, or DAG. Well, all we're going to do is we're going to do this a third time. Right, we add another acyl-CoA, kick off the CoA, and now we finally have our triglyceride. We've added the last fatty acid right here. So we have 3 fatty acids on a glycerol. That's a triglyceride. And again, triglyceride is the commonly used term. It's scientifically correct, but technically it doesn't really describe it, right? We don't have 3 glycerol molecules. More technically, it's a triacyl 3 fatty acids glycerol, right? A TAG. And so these terms are actually going to be interchangeable. The rate limiting step, and let me change color here. I'm going to do this in green. This second step right here, and we're not going to name the enzymes of this. We actually will do that in a future lecture. But this second step right here, going from monoglyceride to diglyceride, that is the rate limiting step. Okay. Now, we're not done because we've made, right, essentially, if I can find my, change this to blue, I think we should be able to see that. So here's the blue. Right, this triglyceride synthesis, again, primarily takes place in liver. It's taking, if you will, all excess calories that have been delivered to the liver. It's a little more complex than that, but for simplicity's sake, excess calories get delivered to the liver, and the liver can convert them all to fat. Right? So we started with a glucose molecule, and we can send part of it down to glycerol, we can send the rest of it down to acetyl-CoA, and we can actually go through lipogenesis, create a fatty acid, right? Through fatty acid synthesis. We can do that. And then we can add it to the glycerol, and we can create a fat. We can take excess amino acids, which can enter in either to the citric acid cycle or into glycolysis. Again, they can both become acetyl-CoA and undergo fatty acid synthesis and return back. Anyway, excess calories converted to triglycerides, fats, by the liver. Well, the liver stores some, but the rest of these it ships out. So when you hear about blood triglycerides and what's being measured, those triglycerides are coming from the liver. Okay? And these, in general, get transported to fat in molecules called VLDL. Right? There's more to it than this, but they've got a shell around it, so it'll be water soluble, a protein shell. And then on the inside, there's some cholesterol. There's also triglycerides. And these triglycerides get delivered to adipose tissue for storage. Okay? So it's not just we made them and we kept them here. No, they're going to get shipped elsewhere in the body, primarily adipose tissue, for storage. So our next step here, and I'm going to go back to the red because I like using it, is lipolysis. Right? So the breakdown of fat. Right? So remember, we're doing catabolism. We started with lipogenesis. That's the one that's not catabolism, right? We were making something. The rest of these pathways that we've done, well, I'm sorry, and then fatty acid synthesis, triglyceride synthesis, that's all anabolism. Now we're starting all of the catabolic processes. Here we're breaking down fat, specifically triglycerides. Okay? Triglycerides, we break them down via lipolysis. So remember, we just started with a triglyceride, a glycerol with 3 fatty acids. What we're doing is pulling those fatty acids off, and then we're releasing those fatty acids into the blood. We also release a glycerol into blood, right? So they're all there. We're primarily concerned with the fatty acids. Once those fatty acids are released into the blood, we refer to them as free fatty acids. Right? And those free fatty acids, by the way, are generally transported in the blood by binding to an abundant protein in the blood called albumin. Now, we think of fat as not soluble at all. It's right, we think of, but there's actually solubility levels. It's not very soluble, so you can actually dissolve a very small amount. And so these free fatty acids go to the blood, and there's a small amount dissolved, but then you start binding some to albumin, and that allows more free fatty acids to come into the blood, and they can bind to albumin. You get the idea. There's some that are always floating free, and there's always some that are bound to albumin. But these are fatty acids in the blood, and that's the way we can transport them and give them to other organs, because they can use them for energy. So what organs would actually store fat that would do this? Well, if I can get over here, the liver does this, but adipose tissue stores way more fat. So at times when we need to release fat for use as energy, we will break it down in the liver, we'll break it down from adipose tissue. And the liver might run out relatively soon. Adipose tissue won't. It'll provide your body energy for a long time. Skeletal muscle will also do a small amount of lipolysis, but skeletal muscle, because it's a tissue that requires a high level of energy, would prefer to use those fatty acids by itself rather than sending them off to some other organ. So they're generally used for muscle contraction. Right, and I didn't mention here, but remember, everything takes place in the cytoplasm unless I tell you otherwise. So here's lipolysis also occurring in the cytoplasm, which makes sense because that's where triglycerides are going to be stored, right? It's not stored in some special organelle, usually, but they're stored out there in the cytoplasm. And here are the steps of lipolysis. Okay. We're starting up top here with our triacylglycerol, or triglycerides. And what we're going to do is we're going to pull one of those fatty acids off the triglyceride and release it. Relatively straightforward. Well, the enzyme that performs this is called hormone-sensitive lipase. Guess why it has that name, right? Because it's sensitive to hormones. Lipase, that tells you it's breaking down fat. Generally, right? Enzymes sometimes you wonder how they got their name, but this tells you exactly what it's doing. And when we say it's sensitive to hormones, this is inhibited by insulin. Remember, insulin is a storage hormone. Lipolysis is not storage. We're breaking down energy. So this process is inhibited by insulin, but it's stimulated by epinephrine. And always think of epinephrine as fight or flight hormone. I'm running a 5K race. I am running away from a bear. I'm climbing the tree. Epinephrine, fight or flight. We need to mobilize energy for those processes. Okay. Well, then we now have a diacylglycerol, or diglyceride. Well, we're going to kick off another fatty acid to form a monoacylglycerol, remember, or MAG, the monoglycerides. Well, the same enzyme actually stimulates this step, and that again is hormone-sensitive lipase. We have the same hormones, of course, because it's the same enzyme. It has the same regulation. This, I'm going to turn to blue again here. This is the rate-limiting step of lipolysis. The second step right here. Pretty easy to get the first fatty acid off, a little bit harder, slows down a little bit getting the second fatty acid off. And I could go ahead and speculate why. Maybe it doesn't quite fit in the enzyme pocket quite as well, but it still works. I don't know. I'm just guessing, but you can imagine some different reasons for that. Well, now we're down here to the last step where we've got the monoacylglycerol. We just kicked off the last fatty acid, and now we end up with glycerol. So our products at the end are the 3 fatty acids and the glycerol. Well, that's what we started with when we started with a triglyceride. There's a different enzyme that catalyzes this last step, and it's called MAG, and it's right here, right? The MAG lipase, it will break that down. Well, remember these fatty acids, they've been released. Well, they're going to be secreted into the blood and they can be transported again by binding to albumin. And here's where we define those free fatty acids, right? Fatty acids circulating in the blood. The glycerol is already generally water soluble, so we don't have to worry about it, right? It can already dissolve in the blood. All right, our next process is beta oxidation, which is the breakdown of individual fatty acids. So imagine now these fatty acids were transported and they got into another tissue and we're going to burn them. And many tissues can burn fatty acids, and it can actually occur in 3 different organelles. We are just going to focus on one process. In liver and skeletal muscle, beta oxidation takes place almost exclusively in mitochondria. It can occur in peroxisome in some tissues, even the endoplasmic reticulum. We are focused solely on the mitochondria. In fact, I almost want you to forget this. It's not going to be asked on the question. The question will ask about mitochondria, right? Here is one of our processes that takes place in the mitochondria, and that's beta oxidation. Again, primarily liver skeletal muscle. The skeletal muscle, again, highly metabolic active tissue, lots of mitochondria. It's going to be able to break things down. Here is our process of beta oxidation. Now, this is a little bit more complicated because we're starting, note up here, this is mitochondrial membrane. So this is the mitochondrial membrane right here. And this direction is outside the mitochondrion in the cytosol. And this direction is in the lumen of the mitochondria. It's on the inside of it. And I didn't draw the double layered membranes. That just confuses the issue here. So remember, we broke down triglycerides outside the mitochondria. That took place in the cytoplasm. So we had lipolysis and we ended up with fatty acids, or maybe those fatty acids came from the blood. In either case, the fatty acids are out here. Well, for beta oxidation, we first add a CoA to that fatty acid. So here we go. Remember an acyl. So now we have our acyl CoA. Well, we need to transport the acyl CoA into the mitochondria, which is where we said beta oxidation is going to take place. We need to transport it in so we can break it down and start to extract the energy. We're going to add a molecule called carnitine to it. So here's the carnitine. It's an organic molecule. It's not a carbohydrate, not a lipid, not, can't remember what I said, not a protein, not a carbohydrate, not a lipid. Right? It's another organic molecule. And we add a carnitine to this acyl CoA. Okay? And this is now called acyl carnitine. This enzyme here called CPT catalyzes that reaction. And you note I've drawn CPT right here in the mitochondrial membrane because it is a transmembrane protein and is there in the mitochondrial membrane. Okay? CPT, the actual meaning of that is carnitine palmitoyl transferase. And we'll come back to that in just a moment. So carnitine palmitoyl transferase catalyzes this reaction. Now we have acyl carnitine, and acyl carnitine can be transported into the mitochondria, where the carnitine is pulled back off. The carnitine is transported back out, and look, it can be recycled. It can be attached to another acyl CoA, transported in again. And so it's a cycle for carnitine. And because the carnitine is required to get the acyl CoA, the fatty acid essentially, into the mitochondria and then comes back out, we call this the carnitine shuttle. Okay. I have that asterisk right up here. Oops, right here. 2 asterisks, right? That's indicated up here. That's the carnitine shuttle. And there are 3 enzymes that are involved with this carnitine shuttle. One of them we saw, that's the carnitine palmitoyl transferase. There's actually 2 other enzymes we are not going to name. Okay? And this is the rate limiting step, not just this enzyme, but this whole process of transporting the fatty acid into the mitochondria. That's the rate limiting step. We have to add carnitine, we have to transport it across, we have to pull the carnitine off. By itself, that's already telling you this is going to take a little bit longer. So that's why it's the rate limiting step. Right, so after we've pulled the carnitine off, we're left with, again, an acyl CoA, a fatty acid with a CoA attached. Well, believe it or not, we're going to add another CoA here and end up with acetyl-CoA. And then we're going to pull 2 carbons off at a time, over and over and over. Okay, because remember this fatty acid with the CoA at the end, a fatty acid can be like 16 carbons. We're going to pull 2 off at a time, so we have 14, 2 and we have 12, 2 and we have 10, 2 and we have 8. Remember, fatty acids are always even number. So we always add 2 at a time, we take off 2 at a time. And so every time we pull off a 2-carbon fatty acid, we get an acetyl-CoA. And we're still left with an acyl-CoA. Right? We've added this CoA in so we can create this acetyl-CoA. Well, we already know what we can do with acetyl-CoA, right? We've undergone beta oxidation. We've broken down this fatty acid into all of these individual acetyl-CoA molecules. And acetyl-CoA can go into the citric acid cycle, which also takes place in the mitochondria. So it's a very efficient way. To burn fat, we break down the fatty acids in the mitochondria, and we can already send them straight into the citric acid cycle. Don't get the impression it's fast. It's not a particularly fast process if you compare it with glycolysis, but we can create so many more ATP in this fashion. In fact, we can create, according to one of my sources, 129 ATP from a 16-carbon fatty acid known as palmitic acid or palmitate. That's a lot more than the measly 2 you get from glycolysis, right? Well, now remember, and this is getting ahead of ourselves just a little bit, but acetyl-CoA isn't stuck going into the citric acid cycle. It can also undergo gluconeogenesis to create more glucose. Say it's been 4 days you've eaten, you have to maintain blood glucose levels. You can break down fat. You can convert it. You can use acetyl-CoA, do gluconeogenesis, and you could make some glucose that way. You could also make amino acids if needed, right? Non-essential amino acids we could make as well. Another thing that can happen, and we will see this in a moment, is they can go into ketoneogenesis. So ketogenesis, even though it says genesis, this is still a catabolic process because this only happens during times of, in humans, extreme catabolism. In article number 3, when we're reading it, it's happening overnight in the mice, and that happens in rodents. Overnight, they start to do this. They start to have gluconeogenesis, they start to have ketogenesis, but in humans, it doesn't kick in quite so fast. We'll talk about this in a moment. But ketogenesis, obviously the formation of ketones, right? Well, we make ketones from incompletely oxidized acetyl-CoA. When do we incompletely oxidize acetyl-CoA? When we're only breaking down fatty acids and getting acetyl-CoA that way, and we don't have any coming in from glycolysis, that's insufficient glycolysis, we end up incompletely oxidizing acetyl-CoA, and they get shunted over into this ketogenic pathway. Right? So again, insufficient glycolysis. Well, when would we have insufficient glycolysis? I should have put these 2 bullet points together, but starvation and a fasted state. And you say, well, what's the difference? Well, a starvation, you might be eating every once in a while, right? Fasted state is you haven't eaten for like 24 hours or something, or a starvation is you're having just a little bit over a long period of time. And that's when ketogenesis starts really to occur. It can also occur in uncontrolled diabetes mellitus, or what we just call diabetes. The problem with diabetes is they can't get the glucose into the cell. If you can't get the glucose into the cell, you can't have sufficient glycolysis. And it is not uncommon, especially in type 1 diabetics, that one of the symptoms is they're losing weight. And they're losing weight because they can't get glucose in. They're just burning fat. But they're also having ketogenesis. And this can result in something called acetone breath. We will see why in a moment, because acetone is actually one of the ketones. Acetone is also a nail polish remover, and so yes, nail polish remover, we make it in our bodies, essentially. And that's because these ketones can come out on your breath. And if you've ever gone without food for like 24 hours, either intentionally or not intentionally, I do this once a month for religious observance, but by the end of it, it's a different stinky breath than you wake up with in the morning, because it's these ketones that are coming out on your breath. They're actually a small enough molecule they can do that. Well, why make ketones in the first place? They're not glucose, they're not fat, they're not lipids. Well, the brain and the heart can both use ketones for energy, and they are the most two important organs to support. And now you're in an extreme fasted or starved state, whatever it is, and your body can continue for a long time to supply energy for the heart and brain. In fact, the heart uses about anything you can throw at it. It can even take lactate and push it backwards and burn it for energy. So acetyl-CoA, remember, when we draw it, we can get it through because of glucose, we can get it because of fatty acids, we can get it because of amino acids. Amino acids, again, can enter into glycolysis, or they can enter into the citric acid cycle. Either way, you can end up creating some acetyl-CoA. Ketogenesis is our other pathway that occurs in the mitochondria. So we had beta oxidation and we had ketogenesis. That was it. Everything else is in the cytoplasm. Well, it makes sense to have ketogenesis in the mitochondria, because if you're sending in these fatty acids to be oxidized, broken down, try and extract the energy, and there's not enough glucose around bringing acetyl-CoA in, we're just going to turn them right into ketones, right? Well, these ketones can be released into the blood, and when we have a detectable level of the ketones in the blood, we refer to it as ketosis, because these ketones are also acids, and we're going to learn one of their names is keto acids. They can start to mess with acid-base balance, and if these ketones become sufficiently high that they are starting to lower pH, making you more acidic, that condition is referred to as ketoacidosis. Some students are always going to be familiar with this, what is called the keto diet. I'm not saying I'm in support of it or against it, but the idea of a keto diet is you're limiting glucose, and therefore you would be in ketosis, or you don't want ketoacidosis, but you would be in ketosis more often, indicating again that you're trying to burn some fat. All right, so here is the ketogenic pathway. Once again, we're starting with acetyl-CoA, right? That's what we mentioned up here, that incompletely oxidized acetyl-CoA will go into ketogenesis, and we're actually going to add two acetyl-CoAs together. We're going to kick out a molecule called CoAsh. We won't go into the details there. This is not biochem. And now we have acetyl-CoA. Well, we're not quite done. We still have one more step that we need to go to, or two more steps that we need to go through. We're going to kick off another CoAsh. And now we have a molecule called, look at this, beta-hydroxy beta-methyl-butyryl-CoA, or more simply HMG-CoA. Here, now here's the H, the M, here's the G. Okay, so this is HMG-CoA. This step right here is performed by an enzyme called HMG-CoA synthase. It tells you what it does, right? It made the HMG-CoA. And so this is actually our, let me double check. I just want to check my notes. Yes, that is the rate limiting step. So let me put that in blue. Here's the rate limiting step. Whoops, right here. That's the rate limiting step. Now let's talk a little bit more about this HMG-CoA synthase, right? So remember, this is taking place in the mitochondria. We have the same enzyme out in the cytoplasm where it performs a different function. It's actually involved with the synthesis of cholesterol, or cholesterol biosynthesis. So different place, same enzyme, different role. And this HMG-CoA synthase, the expression of this enzyme is increased by FOXA2. Now, we've either talked about in lecture already or will talk about in lecture, because I'm recording this for all future years as well. FOXA2 is a transcription factor. And it's a transcription factor that increases ketoneogenesis. Well, remember, transcription factors aren't enzymes, so the way they're doing it is they're creating more of the enzymes that are involved, and in fact, that's what's happening. FOXA2, if you're familiar with this terminology, great. If not, just live with it for a little bit. But FOXA2 will bind to the promoter for HMG-CoA synthase and increase the transcription of it. Therefore, you will get higher expression of HMG-CoA synthase, and this pathway will be able to work much more quickly with more enzyme around. Just a reminder that FOXA2 is activated by glucagon, deactivated or inhibited by insulin. As we talked about, this is another lecture. I'm not going to cover that in here, but just to remind you that FOXA2, remember, in response to insulin, it gets phosphorylated. And when it gets phosphorylated, it kicks out of the nucleus, and therefore it can't work anymore. That's why it's deactivated or inhibited by insulin. All right, so this HMG-CoA synthase has catalyzed this reaction and formed HMG-CoA. Well, we're not done yet. We are going to kick off another acetyl-CoA molecule, and we are going to end up with acetoacetate, which is one of our ketones. Acetoacetate can also become acetone, which is another one of our ketones. Acetoacetate can also become beta-hydroxybutyrate, which is another ketone, but note this one can also go back the other direction and become acetoacetate. These are all known as ketones or ketone bodies or ketoacids. These all refer to the same thing, and it's these three molecules right here. And again, the brain and the heart can use these for energy.